again for calling me, and good afternoon from India to all. I am Professor Shyamapada Mandal from India, University of Gour Banga. This is webinar, and due to COVID-19 situation, we are doing our work mostly from home as well as from department, institute, and doing some presentation. And about today's presentation, the top title is Molecular Docking of Vitamin C and Vitamin D with druggable targets on SARS-CoV-2. We know SARS-CoV-2 causing COVID-19. This is a pandemic and this is on. 2 years already passed, but there are no enough treatment. Different trials are on, and some vaccinations has been done. And in different countries, stage 1, stage 2, and booster dose are going on for the protection against this infection of SARS-CoV-2 causing COVID-19. And about vitamin C and D, we have already know that these two vitamins are very important for all different types of against health disorders, such as vitamin C and vitamin D. Both are responsible for disease prevention as well immune boosting, and vitamin C is known as a vitamin that can cause heal against different types of ulceration or infection. And about vitamin D, this is very effective against infection. It is immune boosting. It prevents coldness or infections, even influenza and SARS-CoV-2. But this is not a drug for treatment for SARS-CoV-2 infection, that means COVID-19 patient. Still, these have some protective role, and this is our study because we will explore our molecular docking study of vitamin C and D, their inhibition property against different types of target proteins found in SARS-CoV-2. So this is an in silico, that means computational study applying different types of bioinformatic tools. And vitamin C and vitamin D, these are ligands in molecular docking study. And these are different proteins, 3CL-Pro, PL-Pro, helicase, methyltransferase, RDRP, and S protein. All these are responsible for different kinds of activity in life cycle. Actually, 3CL-Pro and PL-Pro, these two enzymes are responsible for polyprotein breakdown or degradation, resulting into the formation of 16 different non-structural proteins, including 3CL-Pro and PL-Pro itself. Other than helicase, methyltransferase, RNA-dependent RNA polymerase, that means RDRP, and these five are included in this group, and these are responsible for transcription as well as RNA synthesis of virus to complete the life cycle. And another protein, this is S protein, which is responsible for the structural conformation of the viral particle. And in this study, we have utilized S2 subunit of S protein. This is spike protein, S2 subunit. And we have prepared ligand. For ligand preparation, we utilized tools that will be shown in the next page. Protein preparation, that means these are the proteins or enzymes. Against these proteins, we have docked, or that means these are the receptors, and vitamin C and D ligands, they are docked against this kind of proteins from SARS-CoV-2. Molecular docking, after molecular docking, 3D and 2D interactions were analyzed through bioinformatic tools. Then pharmacokinetic analysis through drug likeness and ADMET property were done through Lipinski's rule of 5, bioavailability radar, and ADMET prediction. That means absorption, distribution, metabolism, excretion, and toxicity. This is the study, the software UCSF Chimera version 1.15. This is the website. For molecular docking study, this bioinformatic tool was utilized, and PyMol for visualization of protein structure or ligand structure, as well as the interpretation of different kinds of interaction between ligand and protein after molecular docking. And this open bubble system, this is a tool that utilizing which we can convert one file type to another file, such as PDB2, PDBQT, or different types of files. And biosphere discovery, this is a very important tool for the analysis of 2D to 3D interaction between ligand and protein. PKCSM and SwissADME, these are freely accessible or publicly accessible tools These are very important to analyze the pharmacological property of any kind of drugs or ligands or phytochemicals for their property in silico system. And RCSB PDB, protein data bank, this is responsible for the storage of different kinds of proteins, wherein we have retrieved 6 proteins here from SARS-CoV-2. And PubChem, this is a repository of different kinds of small molecules, such as here vitamin C and vitamin D, that we have utilized as ligands in molecular docking against different kinds of SARS-CoV-2 protein that we have already mentioned. These are the structure, 2D structure of vitamin C and vitamin D, and this is the number of PubChem CID. With this number, we can retrieve from PubChem. This is the number, and drug bank number. This is drug bank number actually, and this is CID PubChem number. So utilizing these two numbers, we can retrieve such structures from PubChem, and utilizing the drug bank ID number, we can utilize also from the drug bank repository. And these are the smiles of these proteins. Alternately, we can retrieve this smile structure for vitamin C and vitamin D, and then we can convert the structure, or we can build the structure in chimera. So this is the process of retrieval system of 3D structure, or directly we can retrieve in SDF file from PubChem. This is vitamin C and vitamin D. And this is the protein 3CL Pro, chymotrypsin-like protease. This is available from SARS-CoV-2. We have studied against this protein with vitamin C and vitamin D. So this is the structure of 3CL Pro and 6WTZ. This is ID. With this ID, we can utilize, and this is a crystallized structure and 3D form that we have utilized in this docking system, and resolution is 1.9 Armstrong. That means this is a very good structure. Still, its authenticity, structural authenticity was determined through Ramachandran plot. This is the Ramachandran plot. We are seeing that almost all the proteins are located within the red region. That means the favored region, most favored region. And in yellow or light yellow, there are only 26 number of amino acids, and in white region, only 1 number of amino acids is present. So structurally, this is very authentic because there is a very less number of amino acid residue located in this allowed region, and almost 90% amino acid residues are available within the red region, most favored region. So this crystal structure is very important, and this is passed in UCLA SEPS project. This is the 3D 1D interpretation. When this kind of test was passed, then this is a good structure of protein for molecular docking usual. This is PL Pro, papain-like protease. Another protein can be retrieved from the PDB ID. This is 6W9C. This is also a crystal protein, 3D form, and this is actually the NSP, and its resolution is very good, more than 2.7 Armstrong. This is the website, and structural authenticity similarly was done by Ramachandran plot. We are seeing that almost 70% of the amino acids are located in the most favored region. So this is a very authentic structure for molecular docking study. This is the third protein, important protein of helicase, and this is the structure PDB-ID 6ZSL. Its resolution is also good, 1.9 Armstrong. UCLA passed 1D 3D. This is also passed, and its Ramachandran plot analysis shows that 92% amino acids are located in most favored region. So this is very authentic structure for the performance of molecular docking against any kind of ligands. And this is the fourth, methyl transferase, can be retrieved from PubChem PDB protein data bank. This is of SARS-CoV-2, and PDB ID 6W61, and this is the structure, and its resolution is 2 Armstrong, and this is very authentic because we know with this resolution about 90% of the amino acids should be located in the most favored region. Here this is true for this kind of property, and this is a very authentic structure for molecular docking. And similarly, RDRP, RNA dependent RNA polymerase, this is the structure, resolution is 3.3, high resolution is PDB ID is 70ZU, And 92 amino acid residues are located in most favored region. So this is authentic structure for molecular docking. Similarly, this is a different protein that 5 wires which were presented earlier, the non-structural protein, and but this is structural protein. In S spike protein, there are two subunits, S1 and S2. We have retrieved S2. This is the structural configuration of S2 subunit of S protein or spike protein, and its PDB ID is 6LXT, and resolution is 2.9 Armstrong, and 96% amino acids are located within the red region, that means most favored region. So this is very authentic structure for the performance of molecular docking in silico. And before that, in molecular docking, we have to prepare the protein. So if we consider that here different types of molecules and in the form of water or ions are present, and its ligand are already attached when we have retrieved this structure. But during preparation, we have removed from these structures. Only the protein structures are written in UC shape chimera. So this is about the protein preparation, and after that we have prepared grid box, because this is important for the finding of active site or active pocket, and F pocket system was utilized for pocket formation in server. So these are the dimension and center of different proteins: 3CL Pro, helicase, PL Pro, and RDP, and other two are not presented here. These are the actual dimension in Armstrong. And after molecular docking, this is vitamin C. In A, this is 3CL Pro. Vitamin C was docked in 3CL Pro with helicase. This is methyl transferase. This is S2 subunit of spike protein. This is PL Pro, and this is RDRP, RNA dependent RNA polymerase. So after docking, we are seeing that different kinds of amino acids took part in the interaction with the ligand. This is the structure of ligand. All these are three-dimensional structure. This is the structure of vitamin C, and these all are amino acid residues in 3D form. And after molecular docking, we are seeing that the binding energy ranged from minus 5.4 kilocalorie to minus 4.9 kilocalorie. So this is about PL Pro, and this is 3CL Pro. Other values are located in between. So the minus value were obtained in molecular docking process when we have utilized vitamin C for docking as different proteins, which are represented here. And these are the 3D interaction. All green structures, these are the amino acid, different kind of amino acids in all cases. That means 3CL Pro, helicase, methyl transferase, RDRP, PL Pro, and spike protein S2 subunit. We are seeing that in all cases, in the interaction between vitamin C and all the receptor protein of SARS-CoV-2, three or more than three number of hydrogen bonds are formed, and these hydrogen bonds are available in different position with different kinds of residues. If you enlarge this view, we can see clearly. Suppose this is C, and this is 3CL Pro. Sorry, this is 3CL Pro, and this is PL Pro. Here 4 hydrogen bonds, conventional hydrogen bonds are found. Others are van der Waals interaction. These all together create the minus value of binding energy. That means these are the responsible for interaction between vitamin C and different kinds of protein from SARS-CoV-2. And thereby the affinity were established with the generation of binding energy in negative value that are represented. And here we are seeing that beside conventional hydrogen bond, also carbon hydrogen bond is seen with threonine. Similarly, we are seeing that all kinds of bonds may be present in the interaction, but here we are seeing that only hydrogen bond in the form of conventional hydrogen or carbon hydrogen bonds are available. So this is about the interaction between different kinds of SARS-CoV-2 protein and vitamin C, and binding energies like this. And this is another interaction between all kind of proteins of SARS-CoV-2, all 6 kinds that are represented here. and this is vitamin D. This is the structure of vitamin D, all cases. This is 3D structure, and the different amino acid residues are represented here in the interaction. First A to F, this is 3CL pro, this is helicase, methyl transferase, PL pro, spike protein, and DRP. Now we will see the kinds of different bond are formed. We are seeing that only in case of C, D, and E, that means methyl transferase, PL pro, and RDP, hydrogen bonds are available. But in case of others, only hydrophobic bonds are seen. So if you enlarge this structure, we can view this. These are the hydrophobic bonds with different kinds of amino acids. There are no hydrogen bonds like C and E. This is hydrogen bond, this is hydrogen bond, and others are hydrophobic bonds. And the green color, these are the van der Waals interaction. So all kind of bonding or interaction are available in achieving the binding energy. Here, this is with threonine, conventional hydrogen bond is formed, but this is devoid of any hydrogen bond. Only hydrophobic bonds are formed in the form of pi sigma alkylyl or pi alkylyl. So this is about the interaction. This is 2D interaction, and we are seeing that for 3CL pro, when it docked with vitamin D, the binding energy was minus 8.9 kilocalorie per mole. For helicase, this is minus 9.8. For methyl transferase, this is minus 9.2. PL pro minus 9.5, RDRP minus 9.5, and S2 minus 8.7. When we analyze this finding, we know that when binding energy is minus 12, then the A to F. Now, about the ADME and toxicity property through PKCSM or tool, it was utilized Swiss ADME. So, and ADME analysis that will be represented now. This is the property: lipophilicity, size, polarity, solubility, flexibility, and saturation. These are the property. When such property value are located in within the pink region of the bioradar, this is the bioradar that can be formed by utilizing the web server Swiss ADME. So all the values are located in the pink region. So this is very authentic to be absorbed in the intestine. So gastrointestinal absorption is high, but it cannot cross blood-brain barrier. And we are seeing that its bioavailability score is good, 0.56. That means it is orally bioavailable, and it also obeyed Lipinski's rule of 5 without any violation because of low molecular weight, which is less than minus 500 gram per mole. The value is here 170.12 gram per mole. And number of donor hydrogen bond and acceptor hydrogen bond are respectively 5 and 10. These are less than 5 and 10. So we are seeing that about all respects, this vitamin C obeyed Lipinski rule. So it is known already because we already utilize for our purpose when clinicians suggest to use this vitamin. So in this case, we can utilize again infection, but not to prevent or against the, it can build immunity, and somewhat it can inhibit different kinds of protein that we have studied in our molecular docking. But solely it cannot be utilized as a drug for the treatment of COVID patient. Similarly, this is vitamin D. Somewhat we have seen that in one cases in Lipinski rule, we think that the log P value That means this value is exceeded beyond the acceptable limit. That means considering this value, we can suggest that this is also obeying the Lipinski rule, because one violation is acceptable and two or more violations are not acceptable. But this is, we know, this is fat soluble vitamin and D, and vitamin C is water soluble. And its bioavailability score is 0.55, though the lipophilicity and insaturation, these in the form of X log P, and in saturation, the fraction CSP3 value are beyond the pink region. And this is the 3D structure of vitamin C, and this is vitamin C, and this is vitamin C, and this is the bioavailability radar, and bioavailability score respectively are 0.55 and 0.56. And this is last W log P versus topological polar surface area plotting for vitamin D and vitamin C. This is wild egg plot. Vitamin C are located within the white region, and any kind of small molecule are placed with this value within the white region of egg. Then this is gastrointestinal. This is absorbed passively in our gastrointestinal wall. But vitamin D, this is beyond of the white region, so this can be absorbed very slowly. Neither vitamin D nor vitamin C are located within this yolk region, so both are non-permeable to blood-brain barrier. And PGP, about phosphoglycoprotein, both are non-substrate of phosphoglycoprotein enzyme, because these are presented in red color spot or round, and in case of PGP positive, this should be marked with blue color. So all these are PGP negative. And lastly, the highlights: molecular docking authenticated the inhibition of SARS-CoV-2 proteins by vitamin C and vitamin D. Both the vitamins had good affinity to all the target proteins considered. Stable protein ligand complex formation was due to high binding affinities of ligands to the target proteins, especially the vitamin D. Vitamin D was found very effective against all the target proteins from SARS-CoV-2. Admit properties. This is very important pharmacodynamics or pharmacokinetic properties for small molecular characterization that could be utilized as a drug against any infectious or non-infectious disease. So admit profiling signifies the usefulness of the two vitamins against SARS-CoV-2 infection causing COVID-19. But here, vitamin D is very effective in the inhibition of 6 types of proteins from SARS-CoV-2. Still, both the vitamins can be utilized for the building of immunity in our body, and in this way we can prevent or reduce the chance of infection by SARS-CoV-2, because there are no confirmed drugs for the treatment of SARS-CoV-2 infection or COVID-19 patient. Though vaccine are available, but vaccines activity is not permanent. So vitamin C and vitamin D can be taken from our diet. Actually, vitamin C, because this can be found from different leafy vegetables, and vitamin D is synthesized in our skin in sunlight, actually in the morning sunlight. This is very useful, and it can be available from mushroom or other some vegetables and different types of marine fishes. So this is all about vitamin C, vitamin D against SARS-CoV-2 or COVID-19 patient that can be utilized to boost our immunity against SARS-CoV-2 infection. So thank you. I acknowledge second edition of Internal Precision Medicine Conference Virtual Event 2022 Organizing Committee, organized by Magnus Conference, for the recognition of my research and supporting me for presentation. Thank you. Stay safe, stay healthy. Thank you, ma'am.